Hello and welcome to my academy. Today we are going to discuss about the glycolysis. Glycolysis is also known as Embeden-Meyerhoff pathway. It is an oxidative process in which one molecule of glucose is partially oxidized into two molecules of pyruvate in a series of enzyme catalyzed reactions. Glycolysis occurs in cytosol of all the cells. On the cell membrane are present the glucose transporters that transport the glucose molecule. Once glucose enters into the cell, it gets phosphorylated. The phosphate donor is ATP molecule. Thus, glucose gets locked or trapped inside the cell to form glucose 6 phosphate. The process of glycolysis involves the preparatory and payoff phase. Let us discuss the preparatory phase first, in which the ATP molecule is invested or utilized. Glucose molecule is phosphorylated by ATP to form glucose 6 phosphate. This process is catalyzed by an enzyme called hexokinase plus magnesium divalent ion. This process is irreversible. Number second step in the glycolysis involves the conversion of glucose 6 phosphate into fructose 6 phosphate by the presence of an enzyme called phosphofructoisomerase, a reaction in which aldose is converted into the ketose. This reaction is reversible. Number third step in the glycolysis involves phosphorylation, in which fructose 6 phosphate is phosphorylated by ATP to form fructose 1,6 bisphosphate. This reaction is catalyzed by an allosteric enzyme called phosphofructokinase. This reaction is irreversible. Thus, ATP is utilized in this step. Number fourth step in the glycolysis involves the cleavage, in which the fructose 1,6 bisphosphate is cleaved to produce three carbon molecules, that's glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. And this reaction is catalyzed by an enzyme called aldolase. Number fifth step is isomerization. Dihydroxyacetone phosphate is isomerized to form glyceraldehyde 3 phosphate and is catalyzed by an enzyme called triose phosphate isomerase. However, dihydroxyacetone phosphate is rapidly converted into glyceraldehyde 3 phosphate. Now we have two molecules of glyceraldehyde 3 phosphate. Now from here, payoff phase starts. The two molecules of the glyceraldehyde 3 phosphate are oxidized. Enzyme glyceraldehyde 3 phosphate dehydrogenase catalyzes the conversion of glyceraldehyde 3 phosphate into 1,3 bisphosphoglycerate. Here twice NAD positive plus 2 inorganic phosphate gives twice NADH plus 2H positive. Iodoacetate is a potent inhibitor of glyceraldehyde 3 phosphate dehydrogenase. In this step, high energy phosphate group is transferred from 1,3 bisphosphoglycerate to ADP. The formation of ATP is referred as substrate level phosphorylation because the phosphate donor is 1,3 bisphosphoglycerate. This reaction is catalyzed by an enzyme called phosphoglycerate kinase. This reaction is reversible. Now, from these two molecules of 3-phosphoglycerate, we get 2-phosphoglycerate in presence of an enzyme called phosphoglycerate mutase. And the reaction is reversible. Two molecules of 2-phosphoglycerate are converted into phosphenol pyruvate with the removal of water, and the reaction is catalyzed by enzyme called enolase plus magnesium dipositive. In the last step of the glycolysis, the phosphoryl pyruvate gets converted into the pyruvate. Pyruvate kinase is the enzyme involved in the last step of glycolysis. It catalyzes the transfer of phosphate group from phosphoryl pyruvate to adenosine diphosphate to form ATP. Thus, two molecules of pyruvate are obtained from the phosphoryl pyruvate. In the next lecture, we will try to see the fate of the pyruvate. Till then, goodbye. Have a nice day.